Hello, my name is Dr. Bernard Kwabi Ado, a professor of biochemistry and molecular biology and a prostate cancer researcher. I am going to give a lecture describing the biochemical perspective of population health. The objectives of this lecture map to the medical education program for the MD degree in the USMLE Step 1 content categories as follows. 1. To integrate major cellular signaling pathway in biochemistry and pathological contents and predict the consequences of altered function. Apply principles of carbohydrate, protein, lipid, and energy nutrition to metabolism, health, and diseases, and to analyze the clinical manifestations, biochemical consequences, and pathological effects of undernutrition and overnutrition. So by the end of this lecture, you'll be able to explain the public health burden of chronic diseases and their impact on individuals, communities, and different ethnic and racial groups. We'll explain some of the biochemical signaling pathways, principally the phosphoinositol 3 kinase AKT insulin signaling pathway, and how disruption of this pathway leads to insulin resistance. Describe the biochemical mechanisms linking obesity, dyslipidemia, and type 2 diabetes. Analyze how lipid metabolism dysfunction drives atherosclerosis and cardiovascular disease. We want to evaluate how social determinants of health influence biochemical disease pathways, and also be able to apply biochemical knowledge to propose targeted public health interventions, principally disease screening as well as therapeutic intervention. So what is the link between population health and biochemistry? Population health measures provide the information needed to assess and improve community health. This considers infant mortality, social determinants of health, environmental exposures, behavioral factors, and access to health care. At the biochemical level, we are interested in understanding the molecular mechanisms underlying disease by measuring the cellular level metabolic pathways to disease prevalence and be able to come up with molecular targets for drug intervention. We are interested to understand the genetic and epigenetic variation associated with health disparities, as well as biomarkers for screening programs. The most current data on USA mortality rates is from 2024, which shows that the average life expectancy for the US population was 79 years old, and this was an increase of 0.6 years from 2023. The top leading causes of death in 2024 were heart disease, followed by cancer, unintentional injuries, stroke, Alzheimer's disease, diabetes, kidney disease, chronic liver disease, and suicide, making the top 10. These 10 leading causes account for over 70% of all deaths in the USA. When we stratify mortality rates for the top 10 leading causes of death in the USA by race and ethnicity, we see that African American or US blacks are disproportionately impacted by heart disease, cancer, stroke, diabetes, and kidney disease. American Indians are disproportionately impacted by unintentional injuries, whereas Asians or Pacific Islanders have the highest incidence of suicide. So further stratification by age-adjusted death rates by race and ethnicity shows the highest mortality rates among American Indian men, followed by black men, and American Indian or American Native females having comparable death rates as to the white male in USA. Overall, the lowest incidence of mortality was reported for Asian females and males. So what are the factors driving the disparities in mortality rates in the USA? It is believed that we are only scratching the surface of the proverbial iceberg when discussing complex issues driving health disparities. Some of the well-established risk factors associated with health and health disparities include education, occupation, income, access to care, community, culture, genetics, these are specifically genetic variants, gene-environmental interactions, which are captured by epigenetics. Each factor or combination of these factors is believed to be driving health and health disparities. This lecture will focus on the biochemical mechanism underlying metabolic syndrome, which consists of a cluster of interconnected risk factors of metabolic origin that appears to directly increase the risk of cardiovascular disease, type 2 diabetes, strokes, and even cancer, which are amongst the leading causes of death in the USA and global or worldwide. The National Cholesterol Education Program, or NCEP, Adult Treatment Panel 3, has devised a definition for the metabolic syndrome as the presence of three or more of the following criteria. One is central obesity or waist circumference of more than 102 centimeters for men and 88 for women. High triglycerides of more than 150 milligrams per deciliter, high blood pressure, high fasting plasma glucose. This criteria is important for several reasons. One, it allows for the identification of patients who are at high risk of developing atherosclerotic cardiovascular disease and type 2 diabetes. Two, it helps us to better understand the pathological link between increased cardiovascular disease and type 2 diabetes. And three, gives us deeper understanding into the epidemiological and pathological and as well as lifestyle factors. And this allows us to come up with preventative treatment approaches. So I want us to appreciate the roles of chronic inflammation and oxidative stress in chronic diseases. So chronic inflammation and oxidative stress are closely interconnected biological processes and major underlying factors in metabolic disorders and almost all chronic diseases. Chronic inflammation is the pathological condition characterized by combined active inflammatory response and tissue distractions, whereby excess nutrients activate the NF-kappa B pathway, leading to macrophage infiltration and of adipose tissue and increase the expression of TNF-alpha and other pro-inflammatory cytokines, resulting in low-grade systemic inflammation, crosswalk with endoplasmic reticulum stress pathway, ultimately leading to tissue damage. Oxidative stress is caused by excess damaging free radicals released from mitochondria, accumulation of superoxide and hydrogen peroxide, depletion of antioxidant defenses, and acceleration of vascular damage. So when we talk about social determinants of health and health disparities, these have biochemical explanations rooted in nutrition, stress, and environmental exposures. There is mounting evidence to support the causal relationship between many social factors, including the socioeconomic factors and adverse health outcome, not only through the direct relationship, but also through more complex pathway involving biopsychosocial processes. For instance, chronic exposure to social stresses sends signals through the hypothalamic, pituitary, and adrenal, so-called HPA axis, which ultimately leads to increased secretion of cortisol. Environmental stresses induce this so-called wear and tear at the biological level, often measured as allostatic load, whereby excess cortisol release induces physiological changes that causes inflammation and oxidative stress, as previously discussed. This leads to metabolic disorders associated with cardiovascular disease, high blood pressure, obesity, cancer, and other chronic diseases. So what is the role of nutrition in health and diseases? Suboptimal diet is an important preventable risk factor for many chronic diseases. I believe this slide actually ties in with the nutrition lecture from Dr. Yayeng Fang. However, its impact on the burden of chronic diseases has not been systematically evaluated. This Lancet paper carried out a comparative risk assessment approach to estimate the proportion of disease-specific burden attributable to different dietary risk factors among adults aged 25 years and older. Overall, the study identified that diet, high intake of sodium, low intake of whole grains, nuts and seeds, and low vegetable intake, as diet which is high in sugar-saturated beverages, were the leading dietary risk factors for death globally, or at least for the 195 countries that were included in the study. So insulin resistance, as will be discussed in subsequent slides, is a central biochemical link between obesity, type 2 diabetes, cardiovascular disease, and certain types of cancers. The insulin signaling cascade is a crucial biological pathway that regulates glucose metabolism, cell growth, and energy storage. When insulin binds to its surface receptors, it triggers a cascade activation of many intermediates, including the glucose transporters GLUT4, to the cell membrane, and promote the synthesis of glycogen, lipids, and proteins. The insulin receptors activate a complex intracellular signaling network through the insulin receptor substrate protein and the canonical phosphoinositol 3 kinase and the ERK cascade pathways. AKT and phosphokinase B, as well as mTOR, are involved in regulating protein synthesis. In the liver, insulin promotes the storage of glucose as glycogen and reduces glucose output. In skeletal muscles and fat cells, insulin promotes glucose transport through the GLUT4 to be used for energy production and for metabolism. I believe this slide actually ties in with the lecture from Dr. Fedora Sutton. So the discovery of insulin has become a milestone and has truly revolutionized both the therapy and the prognosis of diabetes. Foods that causes the largest insulin spike are carbohydrates, especially refined carbohydrates and sugars. On the other hand, proteins and high-fat diet do not induce appreciable increase in insulin secretion. The importance of insulin in regulating blood glucose was first demonstrated by administering insulin extracts to dogs and later to humans with type 1 diabetes. This picture here shows a 3-year-old boy before and after insulin administration, a study that was carried out in 1923. After just 2 months of insulin treatment, the boy looks healthy and normal, having doubled in weight. So I want to remind you, insulin actually increases weight gain. So insulin plays a role in multiple processes and pathways: glucose uptake, glycogen synthesis, lipolysis, protein synthesis, gene expression, DNA synthesis, sodium and potassium ion channel pumps, apoptosis, and autophagy. Insulin disorders occur when the cells stop responding to insulin, forcing the pancreas to overproduce it or to keep blood sugar in order to keep blood sugar stable. I believe this slide ties in with a lot of the lectures from either Dr. Sutton, Dr. Sharma, Sutherland, and also Dr. Fang. Because insulin is involved in many and multiple biological pathways, its actions need to be carefully regulated to ensure proper metabolic function and to maintain energy balance. Insulin resistance is caused by a disruption in the regulation of glucose metabolism in body tissues such as the liver, adipose tissue, and skeletal muscle. It is generally associated with hyperglycemia, hyperinsulinemia, and hyperlipidemia. Impairment of insulin action is caused by several factors, including hyperinsulinemia, increased adiposity, enhanced inflammatory signals, endoplasmic reticulum stress, an increased expression of adipokines, mitochondrial dysfunction, elevated free fatty acids, and dysfunctional insulin signaling. So type 2 diabetes is one of the most common metabolic disorders caused by a combination of two primary factors: defective insulin secretion by the pancreatic beta cells and impaired insulin sensitivity in insulin sensitive tissues. At the population levels, it's reported that more than 590 million adults are affected by type 2 diabetes globally. This prevalence has doubled since 2000. We see a significant disparities association with type 2 diabetes, as 80% of cases are reported in low and middle income countries. It's also the leading cause of kidney failure worldwide and gives rise to 2 to 4-fold increased risk of cardiovascular disease. At the pathological level, as we mentioned, type 2 diabetes is significantly caused by a declining insulin secretion as a result of dysfunctional pancreatic beta cells. See high chronic hyperglycemia, which gives rise to glucotoxicity, excess fatty acid, causing lipotoxicity in the pancreas, advanced glaciation end products referred to as AGEs, as well as increased oxidative and inflammatory stresses. So in addition to aberrant insulin signaling, lipid metabolism disorders are major contributors to metabolic diseases. Lipid transport and metabolism describe and govern how the body absorbs, synthesizes, and stores dietary and endogenous fats as triglycerides and cholesterol. Because fats are hydrophobic, they are packaged into water-soluble proteins called lipoproteins to navigate through the bloodstream and be distributed to tissues for energy, hormone synthesis, and cellular repair. Dietary lipids or triglycerides and cholesterol are packaged into chylomicron in intestines and transported in the lymphatic system and bloodstream to be broken down to free fatty acids in muscles and adipose tissues. The liver acts as the body's metabolism control center. It metabolizes dietary and excess body fats by packaging them into very low density lipoprotein or VLDL. VLDL transports triglycerides in the bloodstream to supply tissues with energy. As VLDL loses most of its triglycerides, it becomes denser and shrinks into an intermediate density lipoprotein known as IDL, which is further broken down to low density lipoprotein, and this is highly concentrated with cholesterol. High density lipoprotein or HDL acts as the body's cholesterol scavenger. It retrieves excess unneeded cholesterol from peripheral tissues such as the blood vessel walls and macrophages and safely transport it back to the liver for recycling or excretion. This process is called reverse cholesterol transport. HMG-CoA reductase plays a crucial role in cholesterol production. It serves as the main biological target for statin medications and for managing cardiovascular disease. So the adipose tissue is not just a benign energy store organ. It is a vital endocrine organ that secretes numerous hormones and signaling proteins known as adipokines. In obesity, this system becomes dysregulated, shifting the body towards chronic inflammation, insulin resistance, and metabolic syndrome. Several adipokines or biologically active signaling proteins are secreted by adipose tissue to regulate metabolism, inflammation, and energy balance. In obesity, expanding fat mass leads to adipokine dysregulation, triggering low-grade inflammation, insulin resistance, and a significantly high risk of metabolic and cardiovascular diseases. Atherosclerosis is a chronic lipoprotein-driven disease of the arteries. Atherosclerosis is the main risk factor for cardiovascular disease. And as we learned in the early slides, which is the main or the leading cause of mortality worldwide. It begins with endothelial injury, followed by a cascade of events, the accumulation of lipids, fibrous elements, and calcification that trigger vessel narrowing and activation of inflammatory pathways. LDL infiltration and retention in the arterial wall are the crucial initiating events in atherosclerosis. Step 2 is lipid entry that sparks a chronic maladaptation of inflammatory cascade, and this eventually leads to plaque buildup, restricted blood flow, and increased risk of heart disease or stroke. Step 3 is foam cell formation. So foam cells are basically lipid-laden immune cells, primarily macrophages, that form the core of the atherosclerotic plaques. Their unregulated uptake of oxidized cholesterol triggers arterial inflammation, plaque growth, and the eventual formation of a dangerous necrotic core, directly leading to heart attacks and strokes. Step 4 is the formation of a fatty streaks, the earliest visible signs of atherosclerosis. They form when LDL accumulates in the arterial wall, triggering an inflammatory response where macrophages ingest the cholesterol and form more foam cells. The final step is the formation of a fibrous cap and a necrotic core, with smooth muscle cells accumulating beneath the plaque endothelium. These plaques can rupture, causing thrombosis. Do you see any parallel pathways to the blood coagulation lecture? So as we've learned from previous slides, cardiovascular disease is the leading cause of mortality worldwide. According to the World Health Organization report of 2024, 17.9 million people died from cardiovascular disease globally, and yet it is highly preventable disease through modifiable risk factors. Ischemic heart disease and strokes are the leading cause of cardiovascular disease. So cancer is ranked as the second leading cause of mortality globally after cardiovascular disease. However, there are projections that cancer will overtake cardiovascular disease as the leading cause of mortality in many high-income nations. According to the National Cancer Institute 2026 cancer statistical data, it is estimated that 2.1 million people will be diagnosed with this disease, and more than 626,000 people will die from this insidious disease. This slide shows the top cancer incidence and mortality rates for both men and women in the USA. According to 2025 statistical data from the American Cancer Society for men, the number one commonly diagnosed cancer is prostate, followed by lung and bronchial cancers, and then colon and cancer of the rectum. For women, the number one commonly diagnosed cancer is breast cancer, followed by lung and bronchial cancers, and colon and rectal cancer, making the top three. The highest cancer mortality rates in both men and women are lung and bronchial cancers. The risk factors associated with cancer are multivariant factors. Some well-established risk factors include genetics, age, lifestyle factors such as tobacco smoke, alcohol binge, and diet that is rich in ultra-processed foods, sugar-saturated beverages, saturated fats, and high salt intake, as we saw in the previous slides. Sedentary lifestyle, infection, immune dysfunction, organ site, obesity, metabolic syndrome, and several social determinants of health are driving cancer disparities, cancer incidence, and mortality rates. Similar to cardiovascular disease, most cancers are highly preventable by modifiable lifestyle factors. So it is well established that cancer is fundamentally a genetic disease because it stems from DNA mutations which instruct cells to grow uncontrollably. Its strong link to aging occurs because over decades, cells endure continuous wear and tear as the body repair mechanisms and immune defense are weakened by toxic environment, poor dietary choices, chronic stress, and oxidative stress until the body succumbs to cancer, which is initially localized with the potential to metastasize and become malignant disease. So emerging research data links the disruption of gut microbiome to almost all diseases, including metabolic disease, through multiple biochemical mechanisms. Short-chain fatty acids, primarily acetate, propionate, and butyrate, are the main byproduct produced when the gut microbiome ferments dietary fiber and resistant starches. They are critical for human health because they serve as the primary energy source for colon cells, regulate immune function, and strengthen the gut barrier, so-called intestinal permeability. When microbial balance is disrupted, pathogenic bacteria can degrade the protective mucous layers and weaken the tight junctions between cells, leading to increased intestinal permeability, so-called leaky gut. Bile acid metabolism generates signals that regulate lipid metabolism, inflammation, and gut barrier integrity. TMAO, or trimethylamine and oxide, is a compound formed when gut bacteria digest dietary nutrients like choline and L-carnitine, which are then oxidized by the liver. High blood levels of TMAO are strongly linked to increased risks of cardiovascular disease, chronic kidney disease, and systemic inflammation. So I think it's quite interesting to note that 90% of the cells in the human body are not human. Rather, the human body houses over 100,000 trillion microbes, including bacteria, viruses, and fungi, both on the outside and on the inside of the human body. The gut microbiome in particular plays diverse roles in human development, plays a role in brain and bone development, food metabolism, detoxification of environmental exposure, as well as modulating the immune system. In addition, the gut microbiome modulates mood and stress, as well as control appetite. So diets rich in fruits and vegetables have been reported to have many benefits on the maintenance of the intestinal microbiota by providing essential nutrients. Probiotics are viable microbial food supplements that benefit the host by improving the balance of the intestinal microbiota. Prebiotics are non-digestible substances that stimulate the growth and or metabolic activity of selected GI microbes, leading to health benefits. So to reiterate what we have discussed so far, what is the link between social determinants of health and biochemistry? Social determinants of health, such as education, income, and employment, diet, and physical activity, alter your internal biochemistry through gene-environmental interactions. Diet, chronic psychosocial stresses, and environmental exposures trigger hormonal cascades that regulate cellular metabolism. These environmental inputs physically alter gene expression and immune responses, making societal condition entirely molecular. In the healthcare industry, the biochemical knowledge of these diseases have led to the discovery of biomarkers for disease detection and therapeutic interventions, as we'll discuss later on. So I hope by now you appreciate diet as the single most important determinant of health. However, the accumulating scientific evidence points to a diet rich in processed carbohydrates, sugary products, and certain types of fatty acids as being associated with insulin resistance and metabolic disorders. And these underline many of the chronic diseases that we have discussed in this lecture. Our current dietary patterns are ancient diets that have been revised or whose health benefits are now being recognized. The well-established dietary patterns are as follows: Mediterranean diet, vegetarian or plant-based diet, ketogenic diet, diet high in protein, the DASH diet or so-called dietary approaches to stop hypertension, caloric restriction diet, fasting diet, as well as the modern Western diet that is rich in ultra-processed foods, padded with sugars, salt, and fats, and artificial additives. The ideal diet is that which minimizes ultra-processed foods and pathogenic dietary components and maximizes protective dietary components, including organic fruits and vegetables, fiber, and phytochemical oxidants. What can I say about this slide? Simply, we are sitting too much. A sedentary lifestyle is linked to various chronic diseases, which were traditionally associated with tobacco smoke. Let's just get moving. So unlike sedentary lifestyles, there are so many benefits from physical activity. This increases circulation, improves learning and mental performance, prevents dementia, reduces risk of cancer, cardiovascular diseases, and reduces stress, depression, and anxiety. So I'm excited about the advances in basic science and biomedical research that has reached the point where we can detect many diseases using molecular markers in non-invasive biospecimen, such as saliva, urine, and blood. As a basic science research investigating the role of DNA methylation changes in prostate cancer and prostate cancer disparity, I am proud that the field is at the point of detecting cancer based on a panel of DNA methylation marks in circulating cell-free DNA blood plasma. So furthermore, elucidation of the biochemical signal transduction pathway has led to therapeutic breakthroughs by developing drugs or engineering cells to inhibit, activate, or mimic molecular pathways relevant to certain diseases and conditions. Drugs that target specific pathways have been shown to be efficacious in reversing or treating many metabolic diseases. So similarly, pharmacological drugs that target specific receptors or enzymes can halt uncontrolled cell division in different types of cancers, as shown on this slide. So I hope you've enjoyed this lecture, and here are some key takeaways. So insulin resistance is a central biochemical link between obesity, type 2 diabetes, cardiovascular disease, and also cancer. Chronic inflammation and oxidative stress are shared molecular drivers across many metabolic diseases. Population health disparities have biochemical explanations rooted in nutrition, stress, and environmental factors. The gut microbiome is an emerging modifiable factor that is incredibly important, not only in metabolic but in multiple disease prevention. Understanding biochemical pathways and molecular targets is enabling us to be able to have the ability to screen and to provide treatment at the individual and population level. So here are some questions that I would encourage you to go over, and we'll discuss this when we meet in person. So please take time to go over the questions so we can have some dynamic discussion when we meet. Thank you for your audience.